Paulogia and another edition of Ham and Egg News, where we react to Ken Ham reacting to things.
So I was actually down in Florida and flew back last night, and I didn't know that spring was not here, even though it is. And I get out to my car, and in the end I gave up trying to get into my car. It was covered in ice and it sealed up everything, and I got out the de-icer and that didn't work, and so I had to grab my wife's car and race over and do that. So, anyway.
I never get tired of Ken's travel stories and car troubles. They're always so benign. But he tells them like he survived the Titanic.
Hey, if you coming to the Ark soon, or if you're going, studio audience, some of you, how many of you have already been there? A number of you? How many are you going tomorrow?
Anyone?
Bueller.
When you go down there on the third deck, we just put in a brand new exhibit, our big time charts. Have you seen our time charts of history? The good thing about it is I can block out Georgia Purdom. There we are. We're going to keep this up for the whole of the rest of the program. But anyway.
Again.
There we are, again.
Yeah. This one's 15 feet long, I think.
This one's 15 feet long. The other one's 25 feet long.
25?
Ken is just awful to his staff. But I feel like Georgia probably deserves it.
From Science News, we probably won't hear from aliens, but by the time we do, they'll be dead.
This was a serious article.
Yes, it's very serious.
I don't get the joke. Why is that funny?
Our audience clearly got that it was serious by all the laughing they did.
It's a serious article.
Hey, Paul.
Hey, Scientist Mel.
I was just happening on by, and I heard some pretty ignorant stuff.
Yeah, that was Ken Ham.
And since I bust up bad science like a bad guy on a bender in Gotham, I thought I would come in and resolve some of this nonsense that is passed off as legit. Because it is not.
Still, you could have used the door.
Let's start with this little talk that Ken Ham has.
The reason we're not going to find these aliens, they're already dead.
What this article is trying to do is update the Drake equation. And that's from 1961. Frank Drake originally came out with this equation, trying to find the number of civilizations in the Milky Way where the electromagnetic emissions are detectable. It takes into consideration the rate of star formation that are suitable for development of intelligent life, the fraction of those stars with planetary systems, the number of planets per solar system with the environment suitable for life, the fraction of suitable planets on which life actually appears, the fraction of life-bearing planets on which intelligent life emerges, and then we get into the fraction of civilizations that develop a technology that releases detectable signs that they exist, and the length of time such civilizations release these detectable signals into space. Now, what they're saying is, if we had civilizations that were like us within the Milky Way, even after the civilizations died out, we would still get some kind of broadcasting signal from them if they managed to reach the level of intelligent life that we have, where we're able to broadcast. I mean, we've only been broadcasting about 80 years. And so what this article is trying to say, since we're within this galaxy, we should already have lots and lots of broadcast signals at this point. And the thing is, we don't have a lot of these signals right now. And that in itself gives us a little bit of information. But on the off chance that we do suddenly get a signal, the assumption would be made that it is from a planet that is so far from us that the civilization is likely already gone. That was the point of this article.
So we'll just have these ghost waves floating throughout the galaxy.
If that's the ultimate end of them, they're dead. By the time the signals get here, our civilization will be dead, according to the same philosophy, right?
Ken is sitting here making the assumption that by the time we get a signal, our civilization will be dead. That's not entirely true. We could get a signal tomorrow, but the point being the signal that we get would not be from our own galaxy, and we would know that because of the fact we would have already had these signals by now. So that's a bit of a fallacy in itself. We could have a signal come to us next week. It could be next month. It could be any of those factors. He's making the assumption that this also works in reverse, but that's not the case. It's a flawed argument to think that the only way we would get a signal is if we were dead too. That doesn't make sense at all.
And they get millions of dollars in grants for that. Guess what? I wrote a paper to say why we found no aliens. Oh, let's give you millions of dollars so you can search for more aliens.
It makes this note that they're trying to publish negative findings as a means to further dig into this type of question. Why are there no other broadcasting signals? That's still a question that's worth exploring. Oftentimes we find in science, we don't find enough publications about negative results because sometimes that can spark research that can take us into the right direction.
God's son stepped in history to be a descendant of Adam, to be the God-man, not the God Klingon or something like that, right? But to be the God-man. And so it doesn't make sense. There are races of other beings on other planets. It just doesn't fit with what the gospel is all
Christians
about.
need to think about that.
They have to take into consideration that alien life form doesn't have to be humanoid or in some kind of animal form. It can be single cellular life, much like amoebas, bacteria, paramecium. You can look at the tardigrade. The tardigrade can survive the vacuum of space and doesn't need water for 10 years. It can also survive cosmic radiation. So the assumption that we will never find aliens is a flawed assumption because we do have organisms that can survive the harshest conditions of outer space. But they're making this assumption that life is only humanoid, or I'm guessing animals, multicellular, but we can have single cellular life. And there is active research trying to find out more about single cellular life. So these are questions that scientists want to answer.
If you actually type into a search on the internet, Ken Ham says aliens are going to hell, even some of the newspapers
tons and tons of
put that
articles out
headline. I didn't say they're going to hell. I said they didn't exist.
Remember that frustration when you insist that an atheist hates God, Ken. They don't hate him. They don't think he exists.
Weird crystals could grow on Titan and help alien life thrive. So another alien one.
But there is one place that gives us hope. Titan, the largest moon of Saturn. The only other place in our solar system with an atmosphere. A primordial ecosystem just like Earth, seconds before life was born. Liquid methane raining into huge oceans and lakes that we can't swim in, and an atmosphere rich in nitrogen that we can't breathe. Too cold to exist in, fiercely hostile to life as we know it.
Basically tried to, I guess, simulate the environment to figure out what could happen on Titan. And they figured out that there's these like co-crystals that could potentially form, and they think maybe this weird environment would give rise to weird life forms.
Yeah.
Transporter room.
We are in position above Titan.
Really? Fine job, Mr. Sully. Well done.
Titan has a lot of what we call hydrocarbons, sea, and air. Now, Morgan Cable at NASA, she and her colleagues replicated a lot of the atmosphere.
What's Titan like this time of year? What's Titan like? Titan is exactly like this. All the time. It's got a cloud around it so thick nobody can tell what's underneath. Maybe there's nothing there. There's something there.
Between the flooding and the rain, because it rains methane there, and it floods ethane. Methane is a single carbon with 4 hydrogens attached to it, all single bonds. Ethane is 2 carbons with hydrogens attached to them, all single bonds. We know that the basis of life in regards to molecules and elements are carbon, hydrogen, oxygen, and nitrogen. We have those in all kinds of different types of formations. Now, what they found is like this salt-like compound formed quickly, and it stuck around even though it was washed over. Now, why is this important? This particular salt was formed by ammonia and acetylene. Ammonia is important because that's where we get our nitrogen.
Is Titan really interesting?
What does that matter?
It's like reading a fiction book. Titan is like a parallel Earth. Now read this. Tell me if you think this is a parallel Earth.
So there's these thick clouds, we have muddy terrain, we have regular precipitation, but it doesn't have water. It has liquid ethane, methane, and other hydrocarbons. So these hydrocarbons can come together and potentially form some kind of interesting life form, especially when you have ammonia involved, because that's where we can get our nitrogen. So what are amino acids made out of? They're made out of hydrocarbons with nitrogens. Then we have acetylene used in this case on Titan. It's part of that crystal structure that's formed with ammonia. And acetylene is 2 carbon atoms that have a triple bond and a hydrogen on the other side. So it's a very strong strong bond between those two carbons, so it can give a bit of a backbone, even further structure to this ammonia acetylene crystal. Okay?
Go find on the parallel Earth. Yeah, see if you can breathe ethane, methane. It says
Methane.
the existent methane. No,
Methane.
you pronounced that wrong.
You Australians.
Ken is under the impression that life cannot possibly live on Titan. That's a bit of a flawed statement. When we look at microorganisms on our own planet, Nature published this particular article in 2010 called Methane-Eating Microbes Make Their Own Oxygen. So essentially, we have microbes that eat methane and can generate oxygen from that. It's not too far-fetched to say that there could be forms of life, of bacteria, that survive on Titan based on methane alone. So this particular bacteria that we're talking about, the M. oxophora, can survive in mud that would be considered inhospitable to other forms of methane-consuming bacteria, such as the Methylobacter mesophilicum. That particular species of bacteria does not survive as well. Now, the M. oxyphora, that is a heavily oxygen-producing bacterium, and those microbes primarily live on a diet of methane and nitrogen oxides. Now, given it produces oxygen, but the oxygen has to already be present in the atmosphere, or at least in the area attached to a nitrogen. So if we're looking at ammonia, ammonia is where we get our nitrogen source. This particular article within New Scientist essentially talks about how there is liquid water. So we have ammonia that we know exists in Titan's atmosphere. It's very likely that it's found within the liquid water ocean deep under the moon's icy surface. So I'm quoting the article here. So where do we get our oxygen source in this case? We have liquid water there. So if we have liquid water, we have ammonia, we have hydrocarbons, and if methane is abundant, we already have methane-eating microbes, it's very likely that we could have single-cellular life within Titan. It's also important to note that there are other types of bacteria that eat inorganic types of materials in order to metabolize. These are called lithotrophic bacteria. They can eat anything from hydrogen by itself to iron to sulfur. So we're looking at Titan, and we have all of these things available on the planet for these types of bacteria in order to thrive. So we have an oxygen source through water. We have plenty of hydrocarbons, and we have hydrocarbons that are forming with ammonia in order to make crystals. So it's very, very possible that we could have all kinds of interesting forms of single cellular organisms come from that and utilize all of the resources there in the form of methane, ammonia, carbon monoxide. There is even a type of bacteria that eats carbon monoxide. It's called carboxydobacteria. You should check it out. It's pretty interesting. And the end product of it is carbon dioxide. How cool is that? So Ken is a little bit short-sighted here in the fact that we already have bacteria on our planet that eat inorganic things. And they don't necessarily have to have oxygen as a breathable source in order to survive. And many types of this bacteria were around before plants. At least it's thought that they were because of their types of metabolism.
So they say, well, if you can have a way to get carbon, you can have a way to get organic molecules, you can have life. But the key ingredient isn't all these organic, all these, carbon and all that stuff. It's information. You need information for life. You need DNA.
Here's the thing. DNA is made up of a hydrocarbon backbone with a phosphorus that connects the oxygens. Then it has on the inside what we call nitrogenous bases. These nitrogenous bases have what in them? Nitrogen. Since we know that Titan has a water source, liquid water being an oxygen source, and we know it has substantial amounts of nitrogen, who's to say that instead of a phosphorus on that particular backbone, on that sugar phosphate backbone, there's not a nitrogen there that's connecting the two oxygens? We could very well have a similar type of genetic code in that form, but instead of phosphorus in the backbone, we have nitrogen. Now, I feel like this is a little bit short-sighted because we know that RNA is another form of genetic code, and it's just as important as DNA because that's what ribosomes read in order to make proteins. And all ribosome really is a really giant freaking enzyme that's also got more genetic coding on it, to where it just pulls together all of these different amino acids, and then these amino acids form these long chains. Well, since we know that we have acetylene that's forming crystals with ammonia, a key element of amino acids, who's to say that we couldn't have chemical reactions that eventually lead to where these start to form in some kind of organization based on its own genetic code? We already know that we have bacteria that doesn't need oxygen to live. We have tardigrades that can survive the vacuum of space and be without water for 10 years, as well as survive cosmic radiation. We know these life forms can live in what we would call an inhospitable environment for the rest of us, but to say that these life forms are not possible in other planets, I feel is a vast misunderstanding of our own planet and the organisms that live on it, as well as a complete and total lack of imagination.
Okay, we have to get to these next two articles because I love this is about DNA. So Scott Kelly's Year in Space changed his...
Okay, this is where you actually see a scientist get excited.
I was totally geeking
because
out.
She's a geneticist.
Yeah.
Geeking out.
Geeking out
Yeah,
on me,
see?
right? Because it changed his gene expression.
See, we have a geek here.
Can I talk?
This is a real... Hey, kids, this is a real live geek. I want you to see. You've met a geek. This is a creationist geek.
You know, just as an aside, as I'm sitting here listening to this, Ken Ham talks way too much. So he claims to have scientists on the show, and he just keeps talking. And it's just won't stop. And I'm just like, Ken, you say that you have these scientists on your show and you just talking. He just talks way too much. Oh my gosh. Okay, what are they talking about now?
So there's these twin brothers, and they both happen to be astronauts, right? Which is like never happened before. Scott Kelly and Mark Kelly. So Scott Kelly went up into space, and what they did was before and after, and I think even during, they checked his genes and how much they're expressed versus his brothers, because the idea is that because they're identical twins, they have identical DNA sequence, but how that sequence is read and expressed can be different depending on the environments that each of the twins is in. So the expression of quite a few of his genes had changed while he was in space. Most of them came back to normal, but even 2 years later, 7% of his genes are still expressed differently than that of his brother. So space has actually changed his genetic expression.
The gene expression that they're talking about, genes can be turned on and turned off depending on the environment that you're in. If you go to NIH, there is a genetics home reference that you can look up. It's called Can Genes Be Turned On and Off in Cells? This is something that happens. It's called gene regulation. But this most commonly occurs at the level of transcription. This is when the information in a gene's DNA is transferred to messenger RNA. So this happens from signals from the environment or from other cells. They activate various proteins that are called transcription factors. And so those particular proteins will bind to a certain portion of a gene and increase or decrease the level of transcription. So that's what we're looking at in regards to gene expression, whether or not those particular transcription factors, those proteins, are activating the production of messenger RNA. If you're able to control the amount of transcription of messenger RNA, which is the code that makes the proteins that we see, then that process can determine the amount of protein product that's done by any given gene at a time. So when they're talking about the actual genes weren't altered, but the expression was altered, which means messenger RNA is not being made so much in coordination with the generation of the proteins from the twin that was in space. Something that's pretty interesting that NASA is still trying to work out is how it is that the telomeres, those are the things that are on the end of DNA, and what we have whenever our cells undergo mitosis, our telomeres shrink. Well, you have an enzyme called telomerase, or telomerase is what I call it, and I believe it's called telomerase, that goes back and fills in those shrinking telomeres. Now, the twin in space had elongated telomeres, so they got longer than his brothers on Earth. And so scientists at NASA are still trying to sort out how that's possible. But the only factor that they think is really different is the fact that Scott being in space, he had a relatively healthy diet. And so because of his healthy diet, they think that might have had some kind of impact on his telomeres increasing in size. But one thing that they noticed was that the post-flight measurements showed that his telomeres did shorten in about 48 hours of him landing. So the only thing that the scientists really have to go on, they're thinking that his rigorous exercise regime that he had, because since there's no gravity, you have to exercise a lot more than what you normally would, and a restricted calorie type of intake contributed to him having longer telomeres. So that's something that's pretty interesting in regards to that study. Now, the things that have seemed to have permanent changes in regards to gene expression, not change in DNA, but change in gene expression, would be associated with his immune system response. So his immune system is slightly different from that of his brothers here on Earth. The genes that are involved in bone formation, oxygen deprivation, and DNA repair. These particular genes are expressed differently in Scott from space than his brother on Earth, and they think that a lot of the influence on the expression of those genes happened from the stress of being in space. So what they called space genes, what they meant isn't so much that there's genes they never knew about before, but that certain genes were kind of turned off or maybe expressed considerably less than the rest of us on Earth. But only 7% of his gene expression is different from that of his brothers after landing back on Earth, and it hasn't reverted back to pre-flight levels.
They had to get their little evolution bit in. It says all humans share more than 99% of our DNA, more than 98% of our DNA with chimpanzees. They just had to put that in.
It's just the way
it's not
that
98% similarity.
No, it's the way they're counting the differences that lead them to those numbers.
I don't know where she's getting her information.
I do. We did a video recently on Georgia's objection to chimp DNA comparisons. Spoiler alert, skip to the end. Answers in Genesis uses a nearly useless definition of differences, very poor methodology, rely on several redacted studies, and tout a study that leverages a short-lived bug in a software program to get the results they want. Click on the eye above or a link in the description for the full video.
The thing is, when we're categorizing organisms on how closely related they are to one another, it isn't just based on genetic code. It's based also on bodily structures, embryology, biochemistry, how things are metabolized. Even behavioral aspects to a degree are taken into consideration on determining where organisms fall on the tree of life. So it's not just genetics. Now, it is important to note that 2% difference is kind of what we see phenotypically on the outside when we're looking at chimps. That slight variation is still enough to show a profound change. So it is important to note that genetic code is a powerful thing, and slight changes can modify how proteins are expressed in an organism. But just because something looks slightly different from us doesn't mean we're that incredibly different.
This pendulum has swung in this nation. Churches once had a favoured position. Now, because of the secularization of the culture, it's that worldview clash.
I mean, I don't know how anybody watches this without staying angry.
I think Bruce Banner expressed it best.
That's my secret, Captain. I'm always angry.
This is, my friends, what we call bad science. This is pushing a perspective by cherry picking what they want, presenting a particular perspective and saying, hey, look, this is our interpretation of what this says. This is why I don't watch shows like that so often, because I just want to get angry and punch things.
Welcome to my world, Mel.
Paul, I just wanted to hop on here because I was hearing a lot of ignorant, nonsensical types of words. And I just wanted to say, keep on keeping on, my friend. And thank you for letting me come in here and air my grievances.
In case it was somehow unclear, Melody is a science communicator par excellence. You can find her on every social media platform ever created as Scientist Mel, including an excellent YouTube channel where you will learn things you never imagined. Go subscribe now and tell her Paulogia sent you. A huge thank you to my patrons who make the work we do on this channel possible. You can join them for as little as $1 a month, so check out the link in the description for more information. Thanks everyone for watching, and tap the subscription button and notification bell for future science episodes and Ham and Egg news.
Have a super awesome, fantabulous day.
Until next time, later.